So we're talking about osmosis still, and I stopped here because I wanted to show you that there's another way to write this, and it was getting kind of long. So anyway, here's one way to write the concentration of stuff like this. You can just write it in percentages. So instead of having a cell and drawing in the dots of salt or sugar or whatever it is, and talking about where the greater concentration is, this is actually way easier. Where is there a higher concentration of water? Here or here, and it just gives it to you as numbers. So you can tell that's got the same concentration in and out. So about as much water comes in as goes out. So that's called an isotonic solution. So isotonic solution, again, equal concentrations of solutes. So what is the direction of water movement? About the same amount in as out. So no net movement, it's called. Here's another thing that you should learn along with dynamic equilibrium, which means as much goes in as goes out. Net movement means if you count all the water molecules out and you subtract from it all the water molecules going in, it's going to be zero. So no movement either way, even though some goes out and some goes in. So the cell is at equilibrium. And pause this if you need to. Okay, here's the next one. Now, again, it's kind of easier this way than the way I was showing you before. 90% water here, 80% water here. Water goes high to low. Boom, right? High to low. Easy. So this is a hypotonic solution because there's more water on the outside, lower concentration of solutes on the outside. So what's the direction of water movement? High to low, which means in this case in. Here's another one, and again, look at the water, 85% water versus 95% water. Water goes high to low, there's more here, less here. So this is called a hypertonic solution because there's lots of dissolved stuff here compared to here. Higher concentration of solutes on the outside. So what's the direction of water movement? High to low, it's going to go out this time. So here's what a red blood cell is going to look like depending on the environment that it's in. So in this environment, you have about the same amount of dissolved stuff in and out. So about as much goes in as comes out. So that one's called isotonic. So in an isotonic solution, there's no net movement of water. About the same amount goes in as goes out. This water, this red blood cell is placed in a solution where there's not too much stuff dissolved out here. So this is actually a oops, I bet I have it written here. That's a hypotonic solution. And that means water goes from high to low, so there's going to be more water out here going in, not too much coming out. So this, what happens here, if it's enough, cytolysis occurs. So cyto for cell, lysis means break, so the cell bursts. Bummer. This last one, you've got a ton of dissolved stuff in here, out here. So this is a hypertonic solution. When you have a hypertonic solution, that means there's not so much stuff dissolved here, so it's a high concentration of water. Most of the water goes out, though a little bit goes in. So with a hypertonic solution, the red blood cell squishes down. It's called plasmolysis. And don't dissect this word too much. It's sort of like the plasma is getting smaller. So I don't know, the goop inside, I don't know why. I guess I don't know how to dissect that word. But this means that the cell shrinks or shrivels. So that's what happens. Excuse me. That's what happens. So that's what happens to animal cells. Again, if you have red blood cells placed in a hypotonic solution, like distilled water, water goes in, and you have enough water going in that it can cause the cell to break, cytolysis. The cell swells and then bursts. If you put water in salt water, if you put red blood cells or any kind of animal cell in salt water, more of the water will come out, and so it's going to dissolve. It'll go from where there's a lot of water to where there's not so much, and you get plasmolysis. The cell shrinks and shrivels. So guess what you see here? This one looks normal, so that would be an isotonic solution. This one looks like it's all swelled up. It almost looks like a circle. So it would be a hypotonic solution because water would be going in. This one, it's hard to see in these, but this one's supposed to be shriveling. So that would be a hypertonic solution because the water is coming out and it's shriveling. So which one is a hypotonic solution? That's the middle one. which shows animal cells in an isotonic. You can just look at the arrows coming out, going in and out, and going in. So isotonic would be this one, equal numbers going in and out. Which picture shows an animal cells in a hypertonic solution? So with hypertonic, there's a lot of dissolved stuff on the outside, and water tends to come out. So that looks like this. Which picture shows animal cells in a hypotonic solution? So in a hypotonic solution, water goes in like it's showing here because it's going from high, where it's a hypotonic solution, almost nothing dissolved in it, inside, and then it might get so much water inside that it would break. So that one. Where is there a higher concentration of solutes? So again, you got to memorize solute. Hey, Lou, come on in. The water's great. So solute is the dissolved stuff. And so the water molecules are the little blue things and the dissolved things are the red things. So you have a higher concentration of solutes inside the cell. And the higher concentration of water would then be outside the cell. And so water goes high to low, right? So which way does water move? Goes from high to low. And so in this case, what way does water go? It goes from where there's a lot to where there's not so much. So water goes in. Quiz time. Where is there a higher concentration of solutes? Again, the solutes are the dissolved stuff, and it's actually labeled here, so the pink stuff. And the water molecules are these things. So there's more solutes on the outside. That picture didn't really work. There we go. The higher, oops, hold on. So the higher concentration of water, let's see. So this stuff is the water, and so water is going to go from high to low concentration. So there's the higher concentration of water. Water goes high to low. And so it's going to go out. Behavior of cells with or without walls in these situations. So cells without walls, like animal cells and most protists, actually require isotonic environments. So like your body does a really good job of maintaining homeostasis, keeping your tissue fluids, the fluid outside your cells, isotonic, so that you don't have to worry about your cells getting swollen or bursting, or the other way, either plasmolysis. So in a hypotonic solution, or in hypotonic environments, cells without walls tend to swell, and if there's too much water, they may burst. So it may burst with too much water pressure. That's called, again, cytolysis, cyto for cell, lysis to break. Some protists that do not have cell walls have contractile vacuoles, remember those? To get rid of the extra water. And so here's the contractile vacuole squirting out water. And so here you can see a protist. And if you are in a hypotonic environment, fresh water, water is going to go from a high concentration to a low concentration. Water is going to keep coming in, coming in, coming in. And so the point of this contractile vacuole is to pump water out. And if it can't pump water out, the cell will burst. So contractile vacuoles are found in unicellular protists that live in fresh water. The fresh water is hypotonic. Whoops, sorry. The fresh water is hypotonic, so too much water comes into the cell. The cell must pump out extra water in order to maintain homeostasis. That means in order to not burst. You're not maintaining homeostasis if you burst. Plant cells are different. So cells with walls, like plants, bacteria, fungi, and some protists, they actually require hypotonic environments, for most of them anyway, in order to keep turgor pressure. So turgor pressure means keeping nice and crisp. So when water comes in, this thing kind of swells, but it doesn't break because of the cell wall. And so turgid is actually normal. That's the way it's supposed to be. So turgor pressure is water pressure pushing the cell membrane out against the cell wall. So water comes in, you get this pressure, but the water doesn't go out because it's a hypotonic environment, so you actually have more water coming in. But anyway, it creates this high pressure and makes the plant nice and crispy. So these cells become limp or flaccid when they lose turgor pressure. So when the water coming in is the same as the water going out, they're kind of droopy. Plasmolysis is when the plasma membrane pulls away from the cell wall in a hypertonic solution. So we'll do this one in lab if we haven't done it already. So water comes in, water goes out, but water in general, if it's a hypertonic solution with lots of dissolved stuff here, that means you actually have more water here. Water comes out. Here's your cell wall. Here's the cell membrane. It pulls away from the cell wall. And that's plasmolysis. So here's the difference in summary between between animal cells and plant cells. Animal cells should be in an isotonic solution. So isotonic is normal for an animal cell. Here's the animal cell. In a plant cell, isotonic actually makes it flaccid. It's not doing great. It needs more water. But for a plant cell, a hypotonic solution is good. Water comes in, makes it nice and turgid. Not so great for an animal cell. They burst when you have too much water coming in because the difference between animal and plant cells here is the cell wall. These don't have a cell wall to hold them together, and so if the pressure inside gets too strong for the cell membrane, they'll just burst. Nobody likes a hypertonic solution when the water goes out, so you'll see this one in lab. Quiz time. Plant cells. These plant cells must have been placed in which solution? So here's a cell wall. And here's the cytoplasm. This is a purple onion. And so the cell membrane is here. That means enough water has left the cell that this is shrunk down. Water leaves cells when they are in a hypertonic solution. Which animal cell is in a hypotonic solution? So hypotonic is the bad one for animal cells where water comes in and it breaks. So that's this first one. Which plant cell is in a hypertonic solution? Hyper is bad for plant or animal. So that's this last one. That means water's going out. How about an animal in isotonic solution? That's the middle one. It's doing great. Animal cell in a hypertonic solution is the last one there. Not so great. Plant cell in a hypotonic solution. That's the one where the plant is happy. So remember that plants and animals do different in different situations. The animal cell should be isotonic and the plant cell should be hypotonic. How about a plant cell in an isotonic? Here it is. It's not doing awful, but it's flaccid. Not super great for the plant. In a hypertonic solution, plant cells are plasmalized. In a hypotonic solution, plant cells, let's look at it because I'm kind of running through these too fast, I think. Here's the plant cell with the cell wall. Hypotonic means there's not much stuff dissolved out, so there's more stuff dissolved here. So that means there's more water outside. Water comes in, makes it swell, so it's turgid. In an isotonic solution, plant cells are flaccid because you have about the same amount of water going in as out, which means there's not a lot of water pressure in here. In a hypertonic solution, animal cells, let's see, hypertonic means there's a ton of dissolved stuff here. And so water tends to go from high to low, so they tend to shrivel. Close the door, guys. In a hypotonic solution, animal cells, hypotonic solution, that means there's a lot of water outside, not as much inside. They come in. Animal cells tend to burst. Not so good. Isotonic solution, animal cells are just great because you have about the same amount going in as out. Yay for isotonic solutions in animals. Okay.